Hello students, welcome to EPG Pathshala. I am Anup Kumar Kapoor from Department of Anthropology, University of Delhi. Today we are going to talk on module Genetic Markers in Anthropological Research from the paper Human Population Genetics. The objectives are how the types of genetic markers being used evolved in the field of anthropological genetics, then different categories of molecular genetic markers in use, the methods of detection of genetic markers, and the application of genetic markers in answering anthropological queries. Genetic markers, before we start in detail, I would like to tell you that when we talk about genetic markers, there are the three types of main genetic markers. First is the serological genetic markers, which includes AB blood group system, RS blood group system, and other blood group system, right? And there are 35 blood group systems and more than that. Then we also take in genetic markers the biochemical traits, biochemical markers, which includes proteins and enzymes. And the thirdly is the molecular markers, which are the latest one in 21st century. But I will be talking about the latest, that is the biochemical markers in my talk. When we talk about genotyping is an important goal in studying an attribute of interest in genetics. It is used for distinguishing between the genotypes relevant to the variance form of the trait. These variance forms of the trait are also called allelic forms. Genetic markers are harbingers of information on allelic variation at a locus and depend on the mutation process underlying their creation. The three classes of genetic markers available today are allomeres, DNA polymorphism, and DNA repeats. With the advent of cost-effective DNA sequencing methods, the amount of human genetic variation catalog is immense. Genetic markers and the field of anthropological genetics. Genetic markers are genetic entities segregating independently and used to classify populations by their presence, absence, or differences in frequency among populations, as reported by Crawford in 1973. Genetic markers are used to quantify genetic diversity in population that has resulted due to the interplaying of evolutionary processes. The two discoveries of blood group system by Landsteiner in 1900 and that of protein electrophoresis by Smithies in 1955 gave the impetus for genetic marker research. Subsequently, other advancements in methodologies happened, ranging from isolation of genetic material to amplification of selected genomic regions and the reading the composition and the sequence of the genome. All these factors brought in unparalleled flow of information on genetic diversity among human populations described by genetic markers. Genetic markers found application in studying population structure and history, selection, and admixture mapping. This module discusses the different categories of genetic markers that have been widely used to answer queries in anthropological research and their applications in evolutionary genetic studies. Before delving into the molecular markers, a background of the classical markers is, however, important. The alloenzymes, the first genetic markers, the term alloenzymes was obtained from the phrase allelic variants of enzymes. Protein variants of enzymes can be distinguished by separating them in gel electrophoresis according to the size and charge differences caused by amino acid substitution. This is the working principle of alloenzyme markers. The bands showing allozymes variants were visualized by treating the gels with enzyme specific stains that comprise ligands which acted as substrate for the enzyme. Enzyme cofactors and oxidized salt. The use of classical markers for studying genetic variation cannot be underestimated. The reason is that the observation from this group of markers on human population showed immense within population polymorphism, which led to the devising of neutral theory of evolution. The setback that was suffered by this system of genetic markers is the small number of informative loci that could be studied. It had the merit of cost effectiveness and hence was used for genetic mapping of traits in association studies till DNA sequencing became a reality. We have introduction to molecular markers. The alloenzymes markers had the demerit of being indirect way of studying DNA variation and were replaced by direct molecular markers as a consequence of the Human Genome Project. The Human Genome Project generated a human genome reference sequence. This was followed by complete genome sequencing of individuals from different ethnic backgrounds, as reported by Fraser in 2009. The individual sequences varied from the reference sequence at many different locations on the genome. This unleashed the information about different classes of genetic variations. Human genetic variations broadly fall into two categories, that is single nucleotide variants and structural variants. The classes differ on the basis of nucleotide composition, though there is no standard differentiation. Deduction methods. Before elaborating on the types of markers, the different methods available are enumerated in this section. The first is DNA sequence. It is the most precise method available for variant determination. It allows the detection of sequence variants in multiple sequences for multiple individuals. Then second is PCR RFLP. The PCR fragments are digested with a restriction enzyme and the fragment size determined by gel electrophoresis. Base pair substitution at the restriction site lead to changes in the pattern of restriction fragments on the gel. Third is denaturing gradient gel electrophoresis. A double-stranded DNA fragment obtained from PCR is made to migrate through a gradient of denaturing solvents. As the fragment migrates, it gets denatured, leading to a conformational change. The mobility of the fragment is reduced and it reaches a sequence-specific position in the gel. The PCR fragments, which are different in sequence, are characterized by specific denaturation condition. Fourth is temperature gradient gel electrophoresis. The technique is same as that of denaturing gradient gel, only except for the denaturing solvents, a temperature gradient is used for denaturation. The next is single strand conformation polymorphism, that is SSCP. The work principle of SSCP is the difference in electrophoretic mobility of secondary structures found by single stranded DNA fragments. Again, we have the next heteroduplex analysis. In the heteroduplex, DNA molecules are formed by intervening of complementary DNA strands differing at a single or a few bases. Heteroduplex analysis involves assaying differences in the electrophoretic mobility among heteroduplexes or homoduplexes. Then we have the types of polymorphisms. First is single nucleotide polymorphisms, that is SNP. The most prevalent of polymorphisms occur by a single base substitution and is called a single nucleotide polymorphism, that is SNP. In most cases, ASNP, pronounced SNP, has two alternative forms, that is alleles, and as a result transition purine to purine or pyridine to alternative forms, that is alleles, and result of transition, that is mutation or transversion, that is purine to pyrimidines mutation, insertion or deletions, that is in del of a single base or two base also common when the SNP or SNP lies in the recognition site of a restriction enzyme. It is called a restriction fragment length polymorphism, that is RFLP, or a restriction site polymorphism, that is RSP, as the presence of the mutant allele activates the enzyme and results in formation of restriction fragments. The first genetic maps obtained by using DNA polymorphisms were based on RFLP markers. The SNP or the SNP are cataloged in the DBSNP of NSBI and designated with reference to SNP ID. The database is regularly updated as novel polymorphisms are added that are population specific. About 10 million SNPs have been catalogued in double base SNP of NSBI for use in genotyping platforms. Most of the double DBSNP's entries are in DELs, that is 2 out of 12 million entries, as reported by Stachan and Reed in 2003. SNP can either produce an alternation encoding of a amino acid, that is non-synonymous substitution, or code for the same amino acid, that is with the synonymous substitutions. Then comes in the second comes type of polymorphism is mini satellites. Mini satellites are markers composed of tandem repeats of DNA sequences with repeats of length 660 base pairs. Polymorphisms in mini satellites result from unequal crossing or gene conversion genes. The genomic DNA is first digested using restriction enzymes, following which DNA probes containing a complementary mini satellite sequence are allowed to hybridize with the fragmented DNA. The mini satellites are highly polymorphic markers due to their length, and this characteristics has found them application DNA fingerprinting. But their use is limited to forensic investigations and paternity analysis, and this group of markers is not widely used in population genetic analysis. Then we have short tandem repeat polymorphism, that is star RPS, or microsatellites. About 50% of the human genome consists of repeat sequences that are both interspersed and in tandem. Satellites containing tandem repeats, which are 1 to 6 nucleotides long, are called microsatellites. This group of markers form the first PCR-based markers. They are highly polymorphic and widely distributed in the echochromatic part of the genome. These markers are popular in investigation directed at mapping, paternity analysis, and population genetics. Macrosatellites are found by replication slippage, in which DNA polymerase responsible replication of DNA slips and repeats the replication of previous sequences. The markers, however, has complex mutation patterns and create PCR artifacts, complicating band scoring after gel electrophoresis. While SNPs or SNS, SNP are molecular events that have remained stable over evolutionary time, tandem repeats are relatively recent, as reported by Stanton and Red 2003. Interspersed repeat sequences are called short interspersed nuclear elements, that is SINES, means that are 100 to 300 base pairs long or 6 to 8 kilobase long. Interspersed nuclear elements, that is LINES, pronounced as lines. While the satellite markers have been at the forefront of family and forensic studies, the SIN, that is SINE, have been widely used in studying human genome diversity. The ALU family of SINE class has been estimated to be 500,000 copies in humans and is only found in primates. Copy number variations that we call as C and V. These are submicroscopic structural variations on chromosome stretching to more than 1 kilobyte. They vary in the number of copies among different individuals. Although large in size, CNVs are not always pathogenic. This class of variations comprises intermediate size insertions, deletions, and inversions, and large that more than 50 kilobytes segments copy number variations, that is CNVs, as reported by Tuzun et al. 2005. CNVs are due to occurrence of identical or nearly identical sequences of length, that is 1 kilobytes or larger, as reported by Fugue and Scherer in 2006, in some chromosomes, reported by Fraser in 2009. Though SNPs are more common and CNVs account for the greatest number of nucleotides, that is more than 70% of variant bases reported by as Fraser in 2009. They are different between two genomes. The CNVs constitute 0.5 to 1% of the genome of an individual and so act profoundly in evolution of genome and health. A CNV that occurs in more than 1% of the population is called a copy number polymorphism. An inversion forms when a segment of DNA is reversed in orientation with respect to the rest of the genome. A change in the position of a chromosomal segment within a genome in the same or different chromosome, keeping the DNA contact end chain, leads to a translocation. The application of CNVs to study population history has been explored less in comparison to SNPs and microsatellites. Uniparental markers, basing on the mode of inheritance, DNA markers can be classified as uniparental or biparental. Mitochondrial DNA and Y chromosomes markers are uniparentally transmitted. The former is maternally inherited, while the latter is transmitted by fathers. Autosomal markers are transmitted by both the parents. Before a discussion on mitochondrial DNA markers and Y chromosomal markers, one should be acquainted with the terms haplotypes and haplogroups. Combination of allelic states of a set of polymorphism lying on the same DNA molecule, that is, a chromosomal region, is called haplotype. A haplogroup is a set of mitochondrial DNA or Y chromosome haplotypes defined by slowly mutating polymorphisms. Mitochondrial markers. The human mitochondrial genome is a circular, double-stranded, 16,569 base pairs long molecule present in 100 to 1,000 copies in each mitochondrial, as reported by Anderson et al. 1981. The mitochondrial DNA, in form it is written mtDNA, that is mitochondrial DNA, codes for 13 subunits of the objective phosphorylation system of mitochondria, 2 ribosomal RNAs and 22 transfer RNAs, that is the tRNAs. The cellular localization of mitochondrial in cytoplasms enables maternal inheritance, as the egg cells provides cytoplasms for the zygote during fertilization. The mitochondrial DNA has an effective population size of a quarter of that of autosomes, features that are unique to mitochondrial DNA and that have made it to the marker of choice for studying human genomic diversity in general and diversity in maternal lineages in specific are: first, high copy number; second, lack of recombination; third, high mutation rate; and finally, maternal inheritance. The mitochondrial DNA has several SNPs, a subset for which it is also R of LP. Many of the SNPs are restricted to a control region, that is CR, that has a higher mutation rate than rest of the genome, as reported by Stone Aking 2000. Mitochondrial DNA serve as a molecular clock because of the control region that carries genetic signals, that is, promoters, needed replication and transcription. Since much of this DNA segment is not vital to the survival of the mitochondrion or of the host cell, by studying the number and variety of base changes within this control region, geneticists can determine the relatedness between individuals. Using the mutation rate within the mitochondrial control region as a molecular clock, evolutionists can plot the course of evolution. The control region has two segments, that is hypervariable regions. One, positions varies from 1624 to 16,383, and second, 1 to 10,000, which have been widely studied to typify the genetic structure of population. The DNA sequence of a control region is termed hypervariable because it accumulates point mutation at approximately 10 times the rate of nuclear DNA. Most of the studies in which control region sequences have been studied have focused on interspecific patterns of variability and phylogenetic relationship of closely related species, a prominent example being the study of human population history. Certain mitochondrial mutations have also been linked to occurrence of complex diseases as reported by Taylor and Turnbull in 2005. Studies involving mitochondrial DNA variation involved rapid sequencing of PCR products and RFLP analysis of mitochondrial DNA PCR products, but recently sequence analysis of the first hypervariable segment, that is HVR1, of the control region has taken over. There are two basic approaches to using mitochondrial DNA in studies of human evolution. First is linear-based approach, attempts to unravel the history of mitochondrial DNA lineages called haplogroups. Haplogroups represent related groups of sequences that are defined by shared mutations and which tend to be specific for a region. Because haplogroups reflect shared ancestry of mitochondrial DNA, they can be helpful for estimating admixture proportion in populations inhabiting known routes of migration or originating from diverse geographical regions. The second is population-based approach, attempts to study the prehistory of individual populations of geographical regions or of population migration by using human population groups as the unit of study and applying population genetic method to the data. Although HVR1 sequence data alone do not have the resolving power to reveal all haplogroups, the high mutation rate of this segment ensures a sufficient number of polymorphic sites for the population genetics. Then we have Y chromosome markers. The availability of near complete chromosome sequence, thus many new polymorphisms, a highly resolved phylogeny, and insights into the mutation processes now provide new avenues for investigating human evolution. Y chromosome research is expanding. The properties of the Y chromosome that make it to the out tier within the genomic regions are that one half consists of randomly repeated satellite DNA and the rest carries few genes and most of it does not recombine. However, it is because of this that the Y chromosome is used for investigating recent human evolution from a male perspective and has specialized but important roles in medical and forensic genetics. While in a small portion of Y chromosome, the pseudo-autosomal region recombines with X chromosome, the remainder undergoes no recombination. This non-recombining portion of the Y chromosome therefore provides a second DNA region that we can assume undergoes no recombination. However, in comparison with the mitochondrial genome, the polymorphism in Y chromosome is very low. By convention, Y chromosomes are designated to haplogroups or clades. Those that are defined by only short tandem repeats are called haplotypes, and description of data combining both YSTRs and biallelic markers are referred to as lineages. Y chromosome carriers such as diverse spectra of mutation, that is, chromosomal changes that do occur from generation to generation, that can be used at the site or sequence specific markers. As all markers are joined along the entire length of Y chromosome except for the pseudo-autosomal region, a haplotype constructed from a number of different markers actually documents the history of Y chromosome. Though the mutation rate Y chromosome is low and variations are hard to find, rare mutations do occur. Y chromosome haplogroups among world populations. In the Y chromosome haplogroup tree, two primary splits lead to haplogroups A and B, the spread of which is restricted to Africa. Remaining haplogroups form these three subclusters that colons at the root of CRM168 node, representing the majority of African varieties and non-African haplogroups. This level of structure of continental pools of Y chromosome includes the shared presence of haplogroup DE chromosomes in Africa and Asia. The non-African haplogroup C, widely distributed in East Africa, Oceania, and North America, and a global distribution of another non-African cluster haplogroup, that is FM89, with its most prolific daughter group haplogroup K. Considerable regionalization of haplogroups is present in the subclass of F and K. Haplogroups, that is F and KH, are quite restricted to the Asian subcontinent, whereas center of gravity for haplogroups J and I is in Europe and the Middle East respectively. In East Asia, haplogroups, that is N and Q, that arise from the haplogroup K branch are most frequent. Other important K-affiliated haplogroups include Q in Northeast Asia and America, as well as African haplogroup R, whose phylogeography spans North Africa and West Asia and manifests high frequencies in Europe. Now we take the first example as reported by Thangraj et al. 2007. They studied all Austroasiatic populations of India, including the Northeast Khasis. They analyzed the Y chromosome data for 1,222 individuals from 25 Indian populations, covering all subdivisions of the Austroasiatic family. 9 haplogroups were found to be most prominent in this population, with QM95 haplogroup attaining the maximum percentage. Except Khasi, none of the samples showed O2M122. Now we take another example reported by Kumar et al. 1999, surveyed 9 Pakistani subpopulations for variation on the no recombinant region of Y chromosome. The polymorphic site examined were WAP, that is YAP, 5 SNPs, and the tetranucleotide macrocytotide, that is DYS19. Y chromosomes carrying YAP element, that is YAP plus, were found in populations from southwestern Pakistan at frequencies ranging from 2 to 8%, whereas northeastern populations appear to lack YAP plus chromosomes. Autosomal markers. Autosomal markers are inherited from the parents and can hence give more information on the diversity present in the gene pool under investigation. This group of markers gives more representative population genetic information. The autosomal markers undergo recombination unlike uniparental markers and under independent assortment when passed over to the offspring generation. The effective population size is greater than mitochondrial DNA and NRY markers. The genetic markers in autosomal genes under great functional constraints due to the genic richness. That is, autosomes have more genes in comparison to Y chromosomes and mitochondrial DNA and Y chromosomes, and enrollment of genes in metabolism. Mutations in autosomal genes have the ability to have deleterious effect on metabolism and so are under selective constraints. However, some of the mutations have a neutral effect on my phenotype, while others impart advantage in a particular environment. The frequency of markers is hence altered in tune with this effect on the phenotype. The exodus of humans out of Africa and their dispersal in the different continents of the world made them get exposed to varied environment. This included both physical and chemical changes. The metabolic machinery of the Homo sapiens adjusted to the alterations in the environmental leaving genomic imprints in its underlying genes. These genetic traces are now being unraveled to draw inferences on the population history. Demographic changes in history are inferred by modifications that have occurred across the genome, while genetic adaptations are traced by studying signature of selection which are localized in the genome. Some autosomal genes have been widely studied used across human population to answer questions on history of human dispersal. Now, next table shows the some of the genes and their role in metabolism. Now, this table explains the autosomal genes studied in anthropological research, their role in metabolism, and significant polymorphisms that have been studied. When we look at this diagram, we see the name of the gene, for example, the first one APOE. The metabolic importance is lipid metabolism, and the important polymorphism is E2, E3, and E4. Then we take G6PD, that is glycolytic enzyme, again in the RS50308668. All this is written there. Then we have PTC. Now we have the PTC taste perception, and there is no. So means here, let's summarize what we have learned in this module. The first is genetic markers are genetic entities segregating independently and used to classify populations by their presence, absence, or differences in frequency among populations. Genetic markers are used to quantify genetic diversity in population that has resulted due to the interplaying of evolutionary process. Single nucleotide substitution, what we call is SNP, are the most prevalent or polymorphisms and occur by a single base substitution. SNPs can produce an alternations in coding of an amino acid, that is non-synonymous substitution, or code for the same amino acid, that is synonymous substitution. While SNPs are molecular events that have remained stable over evolutionary time, tandem repeats are relatively recent and hence used to study recent events in populations history. Mitochondrial DNA is characterized by features like maternal inheritance, non recombination, high copy numbers, etc., and that have made it the marker of choice for studying human genomic diversity in general and diversity in paternal lineages in specific. Hypervariable regions, first, the position is 1624 to 1600 383, and second is 1 to 10,000 of mitochondrial DNA have been widely studied to typify the genetic structure of populations. The Y chromosome is used for investigating recent human evolution from a male perspective and has specialized but important roles in medical and forensic genetics. Autosomal markers are inherited from both the parents and give more representative population genetic information. For example, APOE, LCD, and G6PD genes are examples of autosomal genes, the diversity in which have been studied to delineate human population history. Thank you.